13.4 Plant Growth Regulators. 13.4.1 Characteristics. The plant growth regulators, that is PGRs, are small, simple molecules of diverse chemical composition. They could be indole compounds, for example, indole 3-acetic acid and IAA, adenine derivatives, for example, N6-furfuryl aminopurine kinetin, derivatives of carotenoids, for example, abscisic acid ABA, terpenes, gibberellic acid GA3, or gases ethylene C2H4. Plant growth regulators are variously described as plant growth substances, plant hormones, or phytohormones in literature. The PGRs can be broadly divided into two groups based on their functions in a living plant body. One group of PGRs are involved in growth promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. These are also called plant growth promoters, examples auxins, gibberellins, and cytokinins. The PGRs of the other group play an important role in a plant responses to wounds and stresses of biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy and abscission. The PGR abscisic acid belongs to this group. The gaseous PGR ethylene could fit either of the groups, but it is largely an inhibitor of growth activities. 13.4.2 The discovery of plant growth regulators. Interestingly, the discovery of each of the 5 major groups of PGRs have been accidental. All this started with the observation of Charles Darwin and his son Francis Darwin when they observed that the coleoptile of canary grass responded to unilateral illumination by growing towards the light source known as phototropism. After a series of experiments, the site of transmittable influence that caused the bending of the entire coleoptile, figure 13.10. Auxin was isolated by F.W. Wendt from tips of coleoptiles of oat seedlings. The Bacani Fullis seedling disease of rice seedling was caused by a fungal pathogen Gibberella fujikuri. E. Kurosawa 1926 reported the appearance of symptoms of the disease in rice seedlings when they were treated with sterile filtrates of the fungus. The active substance were later identified as gibberellic acid. F. Skoog and his co-workers observed that from the internodal segments of tobacco stems, the callus is a mass of undifferentiated cells, proliferated only if, in addition to auxins, the nutrients medium was supplemented with one of the following: extracts of vascular tissues, yeast extract, coconut milk, or DNA. Miller et al. 1955 later identified and crystallized the cytokinesis promoting active substance that they termed kinetin. During mid-1960s, 3 independent researchers reported the purification and chemical characterization of 3 different kinds of inhibitors: inhibitor B, abscission 2, and dormin. Later, all the 3 were proved to be chemically identical. It was named as abscisic acid ABA. H.S. Cousins 1910 confirmed the release of a volatile substance from ripened oranges that hastened the ripening of stored unripened bananas. Later, this volatile substance was identified as ethylene, a gaseous PGR. Let us study some of the physiological effects of these 5 categories of PGRs in the next section. 13.4.3 Physiological effects of plant growth regulators. 13.4.3.1 Auxins. Auxins, from Greek auxin, to grow, was first isolated from human urine. The term auxin is applied to the indole 3-acetic acid IAA and to the other natural and synthetic compounds having certain growth regulating properties. They are generally produced by the growing apices of the stems and roots, from where they migrate to the regions of their action. Auxins like indole acetic acid and indole butyric acid IBA have been isolated from plants. NAA, that is naphthalene acetic acid, and 2,4-D, that is 2,4-dichlorophenoxyacetic, are synthetic auxins. All these auxins have been used extensively in agriculture and horticultural practices. They help to initiate a rooting in stem cuttings, an application widely used for plant propagation. Auxins promote flowering, example in pineapples. They help to prevent fruit and leaf drop at early stages but promote the abscission of the older mature leaves and fruits. In most higher plants, the growing apical bud inhibits the growth of the lateral, that is axillary buds, a phenomenon called apical dominance. Removal of shoot tips, that is a decapacitation, usually results in the growth of lateral buds, figure 13.11. It is widely applied in tea plantations, haze making. Can you explain why? Auxins also induce parthenocarpy, example in tomatoes. They are widely used as herbicides. 2,4-D, widely used to kill dicotyledonous weeds, does not affect mature monocotyledonous plant. It is used to prepare weed-free lawns by gardeners. Auxin also controls xylem differentiation and helps in cell division. 13.4.3.2 Gibberellins. Gibberellins are another kind of promotory PGR. There are more than 100 gibberellins reported from widely different organisms such as fungi and higher plants. They are denoted as GA1, GA2, GA3 and so on. However, gibberellic acid, that is GA31, was one of the first gibberellins to be discovered and remains the most intensively studied form. All GAs are acidic. They produce a wide range of physiological responses in the plants. Their ability to cause an increase in length of axis is used to increase the length of grape stalks. Gibberellins cause fruits like apple to elongate and improve its shape. They also delay senescence. Thus, the fruits can be left on the tree longer so as to extend the market period. GA3 is used to speed up the malting process in brewing industry. Sugarcane stores carbohydrate as sugar in their stems. Spraying sugarcane crop with gibberellins increase the length of the stem, thus increasing the yield by as much as 20 tons per acre. Spraying juvenile conifers with GAs hastens the maturity period, thus leading to early seed production. Gibberellins also promotes bolting, that is internode elongation just prior to flowering in beet, cabbages and many plants with rosette habit. 13.4.3.3 Cytokinins. Cytokinins have specific effects on cytokinesis and were discovered as kinetin. Kinetin is a modified form of adenine upurine from the autoclaved herring sperm DNA. Kinetin does not occur naturally in plants. Search for natural substances with cytokinin like activities led to the isolation of gelatin from corn kernels and coconut milk. Since the discovery of gelatin, several naturally occurring cytokinins and some synthetic compounds with cell division promoting activity have been identified. Natural cytokinins are synthesized in regions where rapid cell division occurs, for example, root apices, developing shoot buds, young fruits, etc. It helps to produce new leaves, chloroplast in leaves, lateral shoot growth, and adventitious shoot formation. Cytokinins help overcome the apical dominance. They promote nutrient mobilization, which helps in the delay of leaf senescence. 13.4.3.4 Ethylene. Ethylene is a simple gaseous PGR. It is synthesized in large amounts by tissue undergoing senescence and ripening fruits. Influences of ethylene on plants include horizontal growth of seedlings, swelling of the axis, and apical hook formation in dicot seedlings. Ethylene promotes senescence and abscission of plants' organ, especially of leaves and flower. Ethylene is highly effective in fruit ripening. It enhances the respiration rate during ripening of the fruits. This rise in rate of respiration is called respiratory climactic. Ethylene breaks seed and bud dormancy. Ethylene breaks seed and bud dormancy, initiate germination in peanut seeds, sprouting of tomato tubers. Ethylene promotes rapid internode or petiole elongation in deep water rice plants. It helps leaves or upper part of the shoot to remain above water. Ethylene also promotes root growth and root hair formation, thus helping the plants to increase their absorption surface. Ethylene is used to initiate flowering and for synchronizing fruit set in pineapples. It also induces a flowering in mango. Since ethylene regulates so many physiological processes, it is one of the most widely used PGR in agriculture. The most widely used compound as a source of ethylene is ethefone. Ethefone in an aqueous solution is readily absorbed and transported within the plant and releases ethylene slowly. Ethefone hastens fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits, thinning of cotton, cherry, walnut. It promotes female flowers in cucumbers, thereby increasing the yield. 13.4.3.5 Abscisic acid. As mentioned earlier, abscisic acid ABA was discovered for its role in regulating abscission and dormancy. But like other PGRs, it also has other wide-ranging effects on plant growth and development. It acts as a general plant growth inhibitor and an inhibitor of plant metabolism. ABA inhibits seed germination. ABA stimulates the closure of stomata and increases the tolerance of plants to various kinds of stresses. Therefore, it is also called the stress hormone. ABA plays an important role in seed development, maturation, and dormancy. By inducing dormancy, ABA helps seeds to withstand desiccation and other factors unfavorable for growth. In most situations, ABA acts as an antagonist to GAs. We may summarize that for any and every phase of growth, differentiation, and development of plants, one or the other PGR has some role to play. Such roles could be complementary or antagonistic. These could be individualistic or synergistic. Similarly, there are a number of events in the life of a plant where more than one PGR interact to affect that event. Example, dormancy in seeds or buds, abscission, senescence, apical dominance, etc. Remember, the role of PGR is of only one kind of intrinsic control. Along with genomic control and extrinsic factors, they play an important role in plant growth and development. Many of the extrinsic factors, such as temperature and light, control plant growth and development via PGR. Some of such events could be vernalization, flowering, dormancy, seed germination, plant movement, etc. We shall discuss briefly the role of light and temperature, both of them the extrinsic factors, on initiation of flowering.